about progression of the disease in different way, more genetic and less, more model focus. I will try to speak or to convince you about the important role of GPCR, homo heteroreceptor complex balance on the Parkinson's disease. But a little of history is not so bad. They could. The idea of receptor-receptor interaction came from, well, the hypothesis was introduced by Professor Fuchs in the 80s, and this hypothesis came from the fact of experiment that they was doing in different membrane preparation from different regions of the brain, analyzing the radioligand binding, using radioligand binding assay, the affinity, the characteristic affinity of different GPCR, and then they found different experimental results that they could not explain in the classical way, thinking in monomers, and they introduced this hypothesis of receptor-receptor interaction. Almost 10 years later, they managed to publish this beautiful paper. At least I get impressed each time that I come back to this figure. They introduced the concept of homodimer and heterodimer, and in this figure they clearly introduced the idea that we have now in the neurons not only monomers, that we was believing in the GPCR field, but also we can have interaction between different protomers, in that case homomers, that they belong to the same subtypes, but also included you can have interaction between different subtypes that they call heterodimer at that time. But the important thing of this figure, there is 2 points here that I wanted to highlight. The first one is that they explain that by this formation for this complex, you can increase the diversity of the signaling in the neurons. It was more easy to explain also all this effect that they was observing in doing pharmacological experiment. But when I was reading carefully the figure legend, I was a little impressed also how so early, I mean we are talking about 1993, they start understanding also that the characteristic of the receptor-receptor interactions will determine the amount of homo versus heterodimer complex in the cells. And this one could have an effect on the signaling, but also now we talk also about the new treatment for the drugs. I mean, early at that time, they understand there could be some kind of balance of population there in equilibrium in some way, and they could be relevant. Not only that, depending on the formation of the complex, you can have different outputs signaling associated. But experimental talking, it takes almost 20 years that people start really opening their mind about GPCR receptor-receptor interaction. That was this paper published, I think so was in Nature by Marshall, and was the first time that using biophysical and biochemical method, they demonstrate the interaction of GABA-B and GABA-B2 protomers. And this interaction takes place via the C-terminal coil structure that form these 2 protomers. It's a special case inside GPCR family. It's not so common to see this kind of coil interaction. It's a strong complex in class A rhodopsin like they are more flexible. The conformation is more, they have more plasticity. But this example was the first demonstration, biophysical demonstration of the interaction between GPCR and GPCR. And of course, after this year, and based on the hypothesis introduced by Fuchs in the 80s, the field started changing completely. People, well, after 2000, there has been some kind of revolution in GPCR field. And a lot of progress has been done. I mean, GPCR is a huge family, it's around 800, but keep in mind that it's only around 300 that they are not orphan GPCR. And a lot of detail has been put in to understand the interface between the different GPCR, I mean, how they could be relevant for the interaction between them or no. Of course, mainly the interface interaction could be, many groups has been focused in the transmembrane because they are hydrophobic, and there are these different kind of model to explain and to determine which interface, which transmembrane could be relevant for the interface interaction. But also, a lot of effort has been put in the electrostatic interaction. It's well known that many GPCR, they have a long intracellular loop and C-terminal tails. These loops or C-tails, they are enriching basic and negative amino acids, forming some kind of clusters, and they help the formation of this complex between GPCR. For example, here in your left, you have a muscarinic receptor M3, and you can see clearly how a strong positive cluster, positive charge amino acid develops in the third intracellular loop and the C-terminal tail. In the case of dopamine D4 also you have a strong positive cluster here formed by lysine and arginine amino acid. But also negative cluster, you can have also the formation of really strong negative cluster. Here in the case of muscarinic 3 receptor, it's amazing how negative is the third intracellular loop. In the case of dopamine D4, it's less obvious the important role of negative charge. Probably it's more a positive charge receptor. And this allosteric interaction, they facilitate the formation of this complex. Also, you should consider the formation of these disulfide bonds. They can cross-talk between them, and they make a more covalent bonds formation of the complex. Interaction between them facilitate the interaction and formation of the complex. Kovilka have got the Nobel Prize a couple of years ago, mainly based in the huge amount of work that he done crystallizing the GPCR in the last year. Keep in mind that until 2006, 2007, we only have the rhodopsin crystal structure, and GPCR is a really huge family, 800 subtype of receptor. After 2006-07, using these special procedures introduced by Kovilka, a lot of novel crystal structure has been developed. And the thing that I wanted to put the attention here or to highlight is all of them has been showing the formation of diamond between GPCR. Of course, that could be an artifact of the crystal structure formation by itself, but combining these results, all of them, there is not even one crystal structure, novel crystal structure, that don't show a formation of dimer, trimer, or tetramer complex of the GPCR subfamily. Combining with the biophysical and biochemical previous data, we can really start getting the idea that there is a common phenomenon for GPCR. This is not something like a liter where some of them are interacting. No, this is quite common. This is an important part on the life of GPCR function and structure. And I just wanted to summarize. I wanted to highlight because there is a lot of concept that has been developed after the hypothesis of Fuchs in 80. And it's important to highlight at the beginning we was really happy with this idea of one neurotransmitter, one GPCR, one G protein, and that is a simple way. But cells is more complex and life systems are more complex. And now we need to really consider that they are not really like monomer structure. They are really forming homodimer and heterodimer, and that could be even more complex with a lot of interacting protein or scaffold protein involved, or another kind of membrane protein like RTK or ion channels that could be interacting or forming part of this heteroreceptor complex. What about the diversity? Well, I was talking now something, I was really mainly describing something more structural function. I was showing that they are forming complex, how they take place, which could be the relevant part of the interface that could be involved or facilitate this complex formation. But the important thing that more relevant for pharmacology could be maybe the diversity introduced by the formation of this complex. Here, for example, in the case of dopamine D2, adenosine A2A, we know that upon coactivation of both receptors in the cell line where they are present, where they exist this heteroreceptor complex, you can see that the coactivation results in inhibition of GIO protein and enhancement of beta-arrestin. Of course, that changed completely the panorama of the signaling of this dopamine D2 adenosine A2A receptor, because now you are enhancing more the internalization. This one is relying on the formation of the complex. This is really a function or a signaling associated with the complex has been demonstrated previously using interacting peptides and knockouts. And what about the specificity? Well, there is a lot of pharmacological guide that they start liking this idea of one neurotransmitter, one receptor, one G protein, and they are also asking all the time, they are wondering, well, it seems that all GPCR are interacting. Yes, it seems, based in the publication of the last 20 years, that all of them in some way form a complex. But this is not really a random interaction. They are quite specific. And I wanted to show last year, in order to show, many people have been looking really in the tree, and they forget to look at the forest, as I mentioned before. Here was the case, many people was focusing one pair of receptor-receptor interaction. And last year we decided to collect all the data that has been published in the last 20 years, and we wanted to put all together. Let me see if this one will be working. Give me 1 second because I cannot see very well from here. Well, we collect all the information. This is human GPCR network interaction. We collect all the paper that has been published. The note here shows the GPCR subtypes, and the edge means the interaction take place between them. And the colors, well, we represent here, based on the family of the GPCR, is the color is a class A rhodopsin light. They are represented by blue, and the secreting light by red, and the metatropic by yellow color. And we collect in human, exactly talking about human subtype, GPCR subtype, we managed to find 183 protomers that has been demonstrated that they are interacting and forming some kind of complex. Here in the network, the thing that I wanted to show, this is the positive interaction. I mean, all the interaction that has been demonstrated using biophysical or biochemical methods that they form complex. And to put an example, let's say dopamine D2, that is quite relevant for Parkinson's disease. Well, it has been demonstrated that dopamine D2 receptor, they can form complex with a lot of different GPCR subtypes. We have dopamine D3, D4, 5-HT2A, we have also dopamine D1, but also we have somatostatin receptor 5 and 2. About the specificity, I promised to talk about the specificity. Well, it seems that dopamine is interacting with a lot of receptors. This is a truth, and the experimental evidence are there, but it's quite specific, the interaction also. If we go to this network that shows GPCR receptor that doesn't interact, I mean this complex that you have there, they are not interacting between them. And we look also for dopamine D2 receptor, we can understand that the dopamine D2 receptor, for example, doesn't interact with adenosine A1, also with muscarinic M4 receptor. I mean, GPCR, they can form complex between them, but this is quite specific. This specificity is relying on the interface interaction, on the amino acid sequence. Some of them are more able to interact with some kind of receptor subfamily or some kind of subtypes, and they don't like also to interact. I mean, that gives an idea of the specificity of this phenomenon. But it's important to keep in mind that in the cells we have all these complex. You have heteroreceptor complex, but also you have the homoreceptor complex, and they are in balance in some way. And it could be relevant for at least for Parkinson's disease, but for all mental disorder disease, to understand this balance. Depending on the pathophysiological conditions of the cells, probably the equilibrium between the heteroreceptor complex versus homoreceptor complex is more disbalanced in some direction or the other way around. And about this one is the thing that I wanted to talk. I bring the example of adenosine A1 D1 in the direct pathway. It has been demonstrated since 2000 that they are forming a complex that was demonstrated using immunoprecipitation and also by physical bioluminescence of fluorescent resonance energy transfer method. Also, it was demonstrated later on that the interaction that takes place between adenosine A1 and D1 receptor is some kind of antagonistic allosteric receptor-receptor interaction. That means activating adenosine A1 receptor, we can block the high affinity state of dopamine D1 receptor, but also we have some kind of negative control or crosstalk at the GIOGS level, intracellular talking. Also, it was demonstrated that adenosine A1 agonists in rabbits can counteract dopamine D1 agonist-induced oral dyskinesia. As I show here, a lot of effort was put in on the heteroreceptor complex in vitro and also in animal models. But I think we need to keep in mind this complexity here. In the neurons, in the cell population, we have, obviously, as has been demonstrated, dopamine D1A1 heteroreceptor complex, but also we have A1A1 homoreceptor complex and the D1D1 homoreceptor complex. In our mind, for Parkinson's disease, to understand the progression of disease, we need to keep in mind this balance that takes place or operate at the cellular level, the membrane level, between these different populations. Also, you have equilibrium between another heteroreceptor complex that could be developed, for example, D1 histamine A3 receptor, D1 mu receptor, or A2A A1 receptor. And yes, it seems quite a little complex, but it's like, well, maybe you remind for the high school in chemistry. Chemical reaction is always equilibrium. Even if it's a long description of the chemical, always the system operating in a direction that enhance one of these, in the case of GPCR heteroreceptor complex, probably in some pathological condition, the equilibrium is more displaced to the homodimer complex, and depending on in other conditions, physiological or pathological conditions, you have more the prevalence of the heteroreceptor complex. And this is the thing that should be discussed in view of the Parkinson's disease. I wanted to enter in more details and to describe how this balance could be relevant to understand the progression of the disease, and also to try to refine the treatment in Parkinson's disease, at least the symptomatic benefits. A lot of dopamine receptor in the direct pathway has been described for dopamine D1, D1D3, D1A1, D1D3A1 has been described that they could be present in the direct pathway and they could be really relevant for Parkinson's disease. For example, in the case I explained before, the formation of this complex developed the activation of A1, developed allosteric receptor interaction with the D1 receptor, counteracting the high affinity state of D1 receptor. And in that case, for Parkinson's disease, what could be relevant? Well, adenosine A1 antagonists could enhance the motor activity of the animal model. Also, the dopamine receptor in the indirect pathway, well, has been described a lot of complex for dopamine D2, mainly dopamine D2, D3, D2, D4, also D2A, A2A that is quite relevant, and the D2A, A2A that has been well characterized and described. For example, in the case of A2A in Japan, for example, the antagonist of A2A is used for the treatment of Parkinson's disease. I will come back later on, maybe we can discuss about that. Even we know now this complex exists in the striatopallidal GABA neurons, and then could be relevant to describe which could be the relevant use of A2A antagonists for the treatment of Parkinson. In view of activation, the agonists of A2A produce a counteraction of the D2 high affinity level. Also, I mean, there is an antagonistic receptor-receptor interaction that operate via the heteroreceptor complex here, and probably you will understand better in the next slide at the pharmacological level how we can understand this concept. But the thing that I wanted to highlight, all these blue GPCR subtypes that I highlighted here, they has been described relevant for Parkinson's disease, mainly D2, A2A, CB1, metabotropic receptor 5, and you can see that all of them can form, they are forming a lot of heteroreceptor complex with different kinds of subtypes. I mean, the diversity of the interaction is quite relevant, and we need to keep in mind this picture of there is a lot of possibility there. But clinical pharmacology, let's talk a little about the progression of the disease and the drug action and the relevance for Parkinson. A lot of people have been discussing here the disease progression, and we can define the disease progression in terms of change in the disease status along the time. And in the case of Parkinson's disease, of course, the natural disease progression is caused by continuous degeneration of the dopaminergic neurons. And regarding the drug actions, well, it reflects the effect of the drug on the disease status. And you can have two possibilities here. One, we can focus the drug actions on the relief of clinical symptoms. I mean, as a symptomatic effect of the drug. But the second possibility is to try to develop drugs that they can really tackle, or they can really block or reduce or make more slow the disease progression, more tackling the molecular basis of the disease, the underlying mechanism of this time course progression of the disease. And for Parkinson, I will come back to the idea of the balance. Well, I show here the progression of Parkinson's disease. It's a quite complex disease, and they develop for unilateral until late stage people start getting dementias. And then the thing that I wanted to borrow is the idea that along all this line of the timelines of the Parkinson's disease, we need to understand, it's important to understand how develop the balance between important heteroreceptor complex for Parkinson, mainly dopamine D2, dopamine D1, adenosine A2A or A1 heteroreceptor complex. We don't know, this is a fusion. We need to really try to understand how this balance is operating. Probably at the beginning of of the onset of the disease, there is more homoreceptor complex of dopamine D2 that you can find later on. And based on that, on this equilibrium, you can optimize or you can define better the treatment, I mean the symptomatic benefit. You can really optimize better the drug treatments and play. Let's say, for example, in the case of D2 adenosine A2A, well, the use of A2A antagonists, if we know exactly along the Parkinson's disease progression how look like the prevalence of dopamine D2 A2 heteroreceptor complex, probably we know better when we can really start giving the combined treatment with L-DOPA and A2A antagonists, instead to start giving probably the A2A antagonists during other stage of the disease that probably is meaningless because there's maybe any, it's not relevant, the formation of the D2 A2A complex there. But it's not only important to understand this equilibrium to get a better feeling of the progression of the disease and how to treat, but also to develop new drugs, for example, depending on the pathophysiological condition of the stage of the disease. Probably we have more heteroreceptor complex coming in, and that makes more sense to start using heterobivalent drugs. I mean, they are the special drug design that they recognize or target the heteroreceptor complex, and we don't need to we avoid the side effects produced by the homoreceptor complex there. Or maybe it will be in some pathological condition that there is a high increase, there is increase in the heteroreceptor complex, that is much better to start disrupting this complex and facilitating the formation of homoreceptor complex counterpart. I mean, if there is, let's say, in some stage of Parkinson's disease, the heteroreceptor complex, the dominant complex there, maybe the development of small interfering peptides or drugs, we disrupt that one and we facilitate the formation of homo complex and can reverse can benefit or can reverse the effect of the disease. About the method, well, it has been taking 20 years to really understand all these concepts of heteroreceptor complex, homoreceptor complex. Now even more we need to start discussing or understanding the balance between them. Always there is a technical limitation. If you don't have the tools to study something, it's meaningless. Maybe you have more or less the concept clear in your mind, but you can move on. And at the cellular model, the use of light resonant energy transfer has been really useful to understand all this complex formation using breadth, FRET, TR FRET. I mean, you can even understand not only to demonstrate if they exist or not, but also the modulation between them. There is a lot of possibility there. But this method mainly you can use in vitro. And in moving more to tissue has been more difficult. Only until the last 3 years it was almost impossible. We can only do radioligand binding assay to understand the allosteric receptor interaction. co-immunoprecipitation also was possible to do in human brain and animal brain preparation. But was the development of in situ proximity ligation assay that allow us to really start characterizing and understanding this complex in the brain, in the tissue. And it's the combination of all, mainly these three methods that will allow us to understand this equilibrium between heteroreceptor complex and homoreceptor complex. I highlight here also the fluorescent cross-correlation spectroscopy. We cannot use in tissue, but in vitro they could be really relevant to understand the balance between the different populations in primary culture cells, let's say, for example, striatal primary culture cells, because the output of this method, this biophysical method, is the movement of homodimer monomers, and monomers, dimer, trimers complex. You can follow and you can understand really the population that you have in the cell model that you are analyzing. At least in vitro, it could be some kind of complementary method. And the in situ PLA, I will come back on Thursday in more details, but I couldn't escape to show quickly, and you can get the idea how it works, and I can show some results in the brain. And the method is really like some kind of a modification of a histological, immunohistochemistry method. You have a primary antibody that they recognize your protomers, your receptor, and then you have a secondary antibody that they are labeled, so they are tagged with oligonucleotides, and these oligonucleotides, they have the property to hybridize, to recognize each other if the distance between them is less than 30 nanometers. And then once they recognize, because the distance is close enough, they form, using a ligase, you can make a covalent link between them, and a polymerase recognize this cycle proof, and you can start really amplifying using polymerase, and you get like a multiplication of this concatenameric oligonucleotide sequence. And at the end, you have a solution with a fluorophore complementary oligonucleotides change that recognize this concatenameric, and you can really follow and observe where the complex has been developed. I mean, the light that you generate here, the fluorescent light that you got, it means it's representative of the interaction of two proteins, or in our case, two GPCR in the tissue or in the brain. I don't know if with the light you can see how look like the dots, but this is different example. For example, this is in the rat brain, the striatum of in the CPU, sorry, yes, in the caudate putamen of CCKB and D2 receptor. This is another example of HT2A dopamine D2 receptor also in the CPU in the rat brain. Here you see the dots coming, the blobs, red blob of PLA, indicating that this complex exists, and they are present in the caudate putamen. And another example, for example, is here in the hippocampus, in the dentatus gyrus, and the CA1, CA3 of the formation of FGF receptor 1. This is fibroblast growth factor receptor with the 5HT1A serotonin 5HT1A receptor. This is a GPCR RTK interaction. You can see all the dots coming, really beautiful expression of the complex in the CA1 pyramidal cells of the CA1, CA3. This is another example. It's just to show the way that with the program you can really map it in the rat brain, the distribution of the complex. This is adenosine A2A metabotropic receptor 5. You can quantify, and you can get the idea how look like the distribution of the complex in the different brain areas. Mainly here we are focusing the heteroreceptor complex, but you can also study or analyze the homoreceptor A2A, or metabotropic 5, metabotropic 5. I bring the example, for example, of A2A. In the hippocampus, you see here the pyramidal cell layer of the CA2, and beautiful distribution, a specific distribution of the PLA positive red blood, only this layer, and you see that in the radiatum is almost absent, really few expression, but it's mainly the distribution up here. It's quite nice to see how in the different region of the brain the complex show different distribution and pattern. This one is the distribution for the A2A and the quantification. I will not enter in details, and this is the example of also in the CA2 of the A1, A2A. You can see that in the same region, you have both homo A2A, homoreceptor complex, and also heteroreceptor A1, A2A complex. Well, it's important to keep in mind that both of them are there, and they are probably in balance. In which direction, I mean, it's more one or the other one, that should be studied and quantified in details. Well, this is important thing to keep in mind. This is more technical thing. I will not enter in details. It's people that is more focused in PLA, the different parameters that you should keep in mind. But the thing that I wanted to highlight here is that taking consideration all these parameters on the PLA method, we can really understand the enrichment or expression of the homo-heteroreceptor complex and start understanding the balance of this population in the brain. For the discussion, well, I think so that we have proposed the molecular phenomenon of receptor-receptor interactions to understand how brain function can increase through molecular integrations of these signals in the brain. And also, it's important to know that all alterations in this specific receptor-receptor complex are indeed considered to have a role in pathogenic mechanisms like Parkinson's disease, and could be relevant to understand during the progression of this disease how they behave or how they appear, and to keep in mind this change in balance with their corresponding homo or heteroreceptor complex. There could be really important field for development of novel drugs. I mean, we can play with the drug that we have already in the market. We can know better which dose we can use, that we can feel in which right moment during the progression of this disease will be relevant to apply a combination of polypharmacological drugs, like for example A2A, D2, L-DOPA with antagonist of A2A, or we can develop novel drugs like heterobivalent or interferon peptides, and also that the PLA will facilitate in the future the understanding of the abundance of this complex in the brains, at least in the rats and mice animal models. Well, now it's time to start discussing around this concept. I mean, receptor-receptor interaction, the relevance for the progression of Parkinson's disease. I think social probably you have more point to discuss, more pharmacological thing probably.
Yes, well, I think it's up to the audience to discuss what they want to discuss. I mean, you have offered a very lovely presentation here, so it's a pity that the light was so full, because we could not appreciate the beauty of these pictures, because it was just so light, we couldn't see it. So, but it actually is quite nice, and you can beautifully quantitate it in a great way, and the quantitation is impressive, and I think with the development of this proximity ligation assay, I think that the majority of neuroscientists, I would imagine nowadays, believe that they also exist in the brain, and not only in cellular models, in artificial systems, but they are there for real. And of course, this has helped us tremendously, because there was a skepticism whether or not they in fact did exist, these complexes within the CNS, in spite of all the strong cellular evidence that has accumulated during the last 20 years and so on. And I guess that when we speak about I will come in with the A2AD to tomorrow in my workshop, so I will not bring it up now. But what I would think because you are interested in Parkinson's disease, and what is the relevance of these complexes then for Parkinson's disease, and Dasiel pointed out very nicely the potential impact of this antagonistic A1-D1 interactions in the direct pathway. You know, the direct pathway, you know about that, the straight to direct GABAergic pathway coming into the ghost pallus interna, to the reticulata of the substantia nigra, and it is an a movement pathway, initiates movements. And then, of course, it is sometimes it's always a matter of in which situation the patient is. So if you want to ask about treatments and so on, just to exemplify, I do believe that things happen in Parkinson's disease. In addition to having A1D1, you have also, in addition, a D3 development in the direct pathway. So you have the D1, D3, which enhance one another. And this is a very substantial enhancement, and they need to be inhibited at least in the case of LDOPA treatment, because I do believe that part of the dyskinesias that develop with Parkinson's disease with L-DOPA and so on is related to an exaggerated activation of this direct pathway through the D1, D3 complex that is not found in the normal brain, but it's formed in the Parkinsonian model brain, at least in the rat, in the hemi-Parkinsonian rat. And then, of course, then you have the option, how do we inhibit it? And then, of course, in that case, to stop these dyskinesias, then if you can develop special A1 agonists that target these D1, D3, and if probably there is an A1, D1, D3 complex, so if you can activate and bring up adenosine levels, so you activate the A1 agonist, you activate with A1 agonist, you can then potentially bring down the dyskinetic behavior, which is of course more complex than that. But I think it could be just one factor involved in the dyskinesias. And then in another case, of course, you have too little activation, then you would like to have preferential A1 antagonists that target these complexes to enhance the D1, D3 signaling. It all depends upon what the other combined treatments. So whenever you discuss these complexes, they do change the treatment, and they do change with the progression of the disease. So it's a very dynamic story, as a matter of fact, and that's why you need to have a much more data in order to optimize potential treatment strategies. So if this is being in development, then whatever has happened in this area now is that the simple thing is just to give combined treatments, right? The problem is, you know, that there are so many complexes, and there are so many A1 or D1 receptors, so you end up with having a lot of side effects. You know, you cannot target it properly. So the old problem of targeting comes up here again. So the trick will be, as you mentioned, the heterobivalent compounds, and or to see, because they should have a special affinity for these complexes, because they bind to the A1, and then they have another through a linker. They have another pharmacophore, and then they have a very high selectivity potential if they can ever enter the brain because they are so large. But we would certainly like to test, at least after CSF injections, to which extent these heterobivalent complexes can, in fact, when reaching at least the brain, can have a potential effect on Parkinsonian symptomatic symptoms. And now, of course, we are aware that we are not going to end up with these treatments, at least not now, to stop the degeneration, stop the progression. We are talking about symptomatic symptoms so far. We are playing on the direct pathway, and we are playing on the indirect pathway, symptomatically, to optimize the treatment, avoid a lot of side effects, and try to tune it so you can match what's going on in that event in the progression of the disease. So that's what we want to do, and I think the future will be, hopefully, that we can also reach into the substantia nigra with these complexities, because these interactions are going on there as well. And in my presentation today, I did involved in producing the enhancement of generation, in addition to all other things, of course. We will take the proteins, aggregating, etc., and mitochondrial problems. But these cytokines released can then produce an enhancement of glutamatic cytotoxicity in cells, including potentially the dopamine cells, because they form a complex between, let's say, interferon gamma cytokine receptor with interaction with the glutamate receptor 1 subunit of the AMPA receptor, and that is mediating the enhancement of glutamatic toxicity. So I think that if you are interested in your information in Parkinson's disease, I think certainly that these pathological receptor-receptor interactions are coming into play, not that you're talking about the symptomatic treatments, but also in terms of, in fact, the degenerative process.
Because, Chef, to be honest, like,
Yeah.
in my mind, I was thinking more at the symptomatic level,
Yes.
in the striatum. And
Yes, exactly.
And
And
I
also I was thinking
I
about that
didn't
too.
know how to correlate, well, at least I didn't know about this paper from the Japanese group,
Yeah, then
that they could be also as part of the molecular base of the
Yes. Yes, I mean,
neurodegeneration process.
certainly.
Binding and everything.
But our work so far has mainly been focused on the symptomatic treatment potential, based on the claim on, I think that to end up is that with the trophic panorama, of course, there is also